Hi everyone, my name is Callie Gustafson and I go by she, her pronouns and identify as bisexual, and I am a sixth year PhD student. And I work under Dr. Laura Gamble at the University of Minnesota. One of the great things about working in the Gamble lab is that we get to use chickens as a system to study development. And very early in development, before a chicken looks like a recognizable chick with its beak and feathers, it actually resembles what a human looks like when we first start to form, where we look like a long column that eventually forms into the spinal cord and brain. And if you were to take a slice out of this column, you'd see that it's actually a tube. And on the back side of this tube, or in this diagram at the top, is where a special population of cells exist called the neural crest. Neural crest cells don't contribute to the brain or spinal cord, but actually end up migrating throughout the body, as you can see here in this video. Their ability to travel is important because that's how they're able to later transform into important parts of your body. So during normal development, neural crest cells eventually can become certain parts of your heart, the neurons in your hands that allow you to sense touch, and even the bones and cartilage that make up the structure of your and even your pets' faces. But when neural crest cells deviate from their normal development, it can lead to birth defects that can change how your heart, skin, or your face develop. So in order to understand how these birth defects happen, we want to know what tools do neural crest cells use to migrate and move through the body during normal development. And one tool that a lot of other cell types use when moving are called exosomes. Exosomes were once thought to be nothing more than tiny garbage bags spit out from cells. They're very small, only about 30 to 100 nanometers in size, which, to put in perspective, the average width of a human hair is about 60,000 nanometers wide. But research has now shown that they actually carry important messages between cells in the body using material like DNA and proteins. And since other cell types have been shown to use exosomes, the hypothesis of my project is that neural crest cells use them too to communicate with one another during migration. One of the great things about working with chicken neural crest cells is that I can take a piece of the neural tube out of the chick, and before the neural crest cells start to migrate, I can put it in a dish and the cells can crawl out onto it. So normally when neural crest cells migrate in a dish, they make these nice big halos that fill up the whole image, which is seen here, stained with a bunch of fluorescent markers to look at different parts of the cell. But when you block the ability of neural crest cells to send exosome messages, fewer cells are able to migrate, and they don't fill in the same area with a nice halo, which is then quantified in these graphs shown to the right. So apart from birth defects, why is it important to study exosomes and neural crest cells? Well, on a molecular level, neural crest cells behave very similarly to cancer cells when they metastasize and spread throughout the body. So the long-reaching goals of my project are to learn what are in these exosomes so I can find out what kind of messages they're sending, and also to better understand how the exosomes are influencing neural crest migration. So not only will this help us better understand how neural crest cells behave and develop, but can also have implications in better understanding cancer cells and how they move too.